everyone. This is Ben Dichter, and I'm going to be going through the tutorial for optical physiology for MatNWB. So in this tutorial, we're going to just generate some fake data for a hypothetical optical physiology experiment of a freely moving animal. And the types that we're going to add to this NWB file are subject, information about the subject, such as species, strain, age, etc. A data type representing the animal's position over time. This could be either their actual physical position or it could be a virtual position in a virtual reality maze. Trials stores information about the trial structure that the animal may be going through. We'll show you how to store acquired two-photon images within and outside of the HDF5 file. We will show you how to store ROIs in an image segmentation object. And finally, how to store the time traces of the fluorescence of those ROIs. So these are just some of the most common data types that we expect people to use in optical physiology experiments. But of course, there are many more types of data that you may need for your specific use case. And at the end, I'll show you some resources you can use to find additional information about other data types. But for now, let's just try to understand how to write an NWB file using these common types of data. So the first thing you're going to need to do is install MatNWB. And I have this already installed, so I have commented out this code. But if you have not already installed MatNWB, then go ahead and uncomment these lines and run this code. So the first line here, the exclamation point makes this a shell command, and this git clone actually gets all of the code for MatNWB. Then we cd into that directory, and now this command recursively adds all of the directories in your current working directory to the MATLAB path. And finally, we have this command here, this generate core command. And to understand what this command is, you need to understand a bit about how MatNWB works. So the way it works is that MatNWB actually reads the NWB schema and generates MATLAB classes based on that schema. It actually writes MATLAB code based on the formal NWB schema. So to tell MatNWB to read that code and read the schema and write those classes, you use this generate core command. If you don't use this command, you won't have a lot of the classes that we're going to be using in this tutorial. So go ahead and run it. It should only take 1 or 2 seconds. If you're having issues with this, try making sure that you're running it from the MatNWB directory as your current working directory. Sometimes that solves some installation issues. So once they have that installed, we can go ahead and start creating our NWB file. So the first thing we're going to do is use this NWB file command to create our root NWB object. And in using this command, we're creating the NWB file object, which contains some session level information about the NWB file. So the whole goal of NWB is to be able to package all of your experimental data in one file that you could give to someone else and they could immediately understand what was going on. So in that spirit, here is some general session level metadata that you might want a secondary user of this data to know about the data. So there are 3 required fields, which are session description, identifier, and session start time. Session description and identifier are free text fields, and session start time takes a date time object which indicates the exact start time of the experiment. So in addition to those 3 required fields, you have a number of optional fields, and I'm showing here information about the experimenter, the session ID, the institution, and related publications. So you'll notice here we're using a key value syntax for entering these variables, and we're going to be using that for most of the classes in MatNWB. So the key here is session description, and the value for that key is mouse in open exploration. So first we're going to create an object representing the subject-specific information. Now for subject, you can have the subject ID, age, description, genotype, sex, species. All of these are free text fields, and you can enter just about anything and it'll be a valid NWB file. But we do have some recommendations for how you can enter this text such that this will be more interpretable by tools that are integrating with NWB. So for age, we recommend using the ISO 8601 duration format. And I'm showing that down here. The subject ID is just 001 as a string, and then the age is P90D, and this means period of 90 days. And you can check out this documentation for how to do other types of durations. Then for species, we recommend using the Latin binomial name. So instead of mouse, you would say mus musculus, or instead of human, you would say Homo sapiens. And for sex, we would recommend using F, M, U if it's unknown, and this happens often, particularly if you're converting old data that doesn't have this information, and O for other if it doesn't fit into those categories. So in order to add the subject information, you create this subject object, and then you add it to the NWB file with nwb.general_subject equals subject. So I'm just going to go ahead and run these cells. Now you see when I run this NWB file constructor, I print it out using this NWB, and you see this nice printout that contains some of the fields that we've populated here, but you can see that there are a number of empty fields that some of which we're going to populate through this tutorial. And then I'll create and add the subject object, and because I didn't terminate this with a semicolon, you can see a nice printout of subject. And you can see that there are a few fields that I've left missing: date of birth, which you can supply instead of age, genotype, and weight. So these are optional fields that you can supply if you know that information. Okay, so now that we have the subject, let's go on to adding behavior information. So there are many special types in NWB for storing specific types of data. And if you want to store position data of a freely moving animal, you should use a spatial series class and a position class. So spatial series is a subclass of time series. And time series is a common base class that is used to measure many different types of time series in NWB. And we're depicting that relationship here. We have time series, which is our general class, and that is extended by spatial series. And what that means is that spatial series is a special case of time series, and it means that all of these attributes that are in time series are also in spatial series. So spatial series has name, description, data, and the units of the data, in this case meters. And it also has a way of specifying the time of the samples. And you can specify regularly or irregularly sampled data. So if you have regularly sampled data, you can use rate and starting time. And if you have irregularly sampled data, then you can express all of those samples as timestamps. And these both would be with respect to the timestamp start time or the session start time. In addition, a spatial series, well, first of all, it specifies that the unit is meters, and also it contains a reference frame attribute, which indicates what the 0, 0 coordinate of the data corresponds to in the physical environment. Now, these spatial series objects are going to be placed within a position object. These position objects can have one or more spatial series. And this kind of indicates that the spatial series is the position of the animal. we are going to also create a processing module called behavior that we can put this data in. So first of all, let's create our position data. Here I'm just generating some fake position data. And I'm using this class, types.core.spatial series, and I'm indicating the values, again, as key value pairs. So we have data is this position data that we just generated. Reference frame, I'm putting in the string 0, 0 is bottom left corner. And here I'm choosing to specify the times of the samples with timestamps. Now here I'm, of course, using a regularly sampled timestamp. So I suppose in this case, I could have used a starting time and a rate, but in this case, I just want to illustrate how to use timestamps. Okay, now that we've created this spatial series object, we can create our position object and put spatial series in it like this. And this sets the name of the spatial series to this string spatial series, and the value of it is this object that we've just created. Now, NWB differentiates between raw acquired data, and this is data that is directly from the acquisition system and should never change, and processed data, and this uses some pre-processing algorithm to create some downstream data from the raw data that you got from your acquisition system. So in this case, we're going to assume that the animal's position is recorded from a video that's monitoring the animal, and this is the result of some video tracking algorithm. So in this case, the position is going to be processed data. Since processed data is very diverse, NWB allows you to create processing modules, which allows you to organize the data based on what type of data it is or what algorithm it came from. So in this case, we're going to create a processing module called behavior. Here is the code for creating that processing module, and we need to give it a description. And now that I've created it, I can use this code to add the position object that we've created into the behavior module. So behavior mod.nwbdataInterface.set, here we're setting the name of this to position, and then this is the position object that we've just created. And finally, we can add this processing module to the NWB file with this code, nwb.processing.set. Here we're specifying the name of this module as behavior, and we're indicating here that this is the module to add. And I'm going to go ahead and run some of these lines. All right, so now that we have some data in our file, let's go ahead and write that file. To write the file, you simply do NWB export, take this object that we've been building, and specify the file path. Okay, so now that's written. Now, how do you inspect the file? So you can simply run NWB read and supply the file path. And I'm going to put this in an object called read NWB file. Now this object looks a lot like the object that we've been using to save this data, but this is the result of reading the file. And if you want to inspect the data inside, you can traverse this hierarchy of the data elements that we've been building. So you can say read NWB file, .processing, get the behavior module, get the position object within the behavior module, and then get the spatial series within that. And here I'm printing that out, and you can see that this has in fact been saved in the read NWB file object. Another way that you can inspect the NWB files is by using the HDF-View tool. So NWB files, as a backend, we are using HDF5 as our primary backend, which means that all of the NWB files are in fact HDF5 files. They're just kind of specific forms of HDF5 files. So you can use the HDF-View tool, which is provided by the HDF5 group, to visually inspect these files. And here I've loaded in the file that we've created, and I'm going into processing, behavior, position, spatial series, And here I've double-clicked on the data element, which brings up this window, and you can actually see the data that we've written into the file. And you can also see some of the attributes, such as meters, of this data set. Now I'm going to show you how to add trials to the NWB file. So trials is an instance of the time intervals class, and time intervals extends the dynamic table class. Dynamic table is a general class that's used to store tabular data, and it's used throughout NWB. So in this case, we're using it to store trial information. It's also used to store electrodes, to store sorted units. We're going to see it later in this tutorial to store information about ROIs. It offers a lot of flexibility for storing tabular data because it allows you to specify required columns, such as, in this case, start time and stop time are going to be required columns for a time intervals table. It also allows you to specify optional columns, and it allows you to specify custom columns, and that's indicated here. You can specify 0 or more custom columns that have their own name and description. As I said, this is tabular data, and you can think of the resulting structure like this. We have our IDs that are required by any dynamic table, and those just indicate the different rows of the table. And then we have start time and stop time, and these are going to be floats that are with respect to the session start time. And you can have any number of optional columns. So in this case, we're going to add one optional column, and we're going to call it correct, and we're going to make it have Boolean values. So here's how you create the trials table, which is a time intervals object. So we're going to use types.core.timeintervals. We need to specify the columns. So here we're going to use start time, stop time, which are both required, and correct, which is our optional column that we're adding. We're going to specify the description, which is required by the dynamic table object. And we also need to specify the IDs. So we're going to use types.hdmfcommon.elementidentifiers. And here we're going to specify that the data is 0 through 2. So here we're using 0 indexing. MATLAB users might not be accustomed to doing that, but here we're going to start counting our rows from 0. And this will make the files that we generate identical to the files that PyNWB is generating in Python. So for the start time, we're going to use a vector data object, which is also in the HDMF_common category. We specify the data and as these float values, and we also specify the description for this object. Columns of dynamic tables are generally vector data. That's kind of the standard type for a column of a dynamic table. So we're going to do the same thing for stop time. We're going to create a vector data with a data field and a description field. And we're going to create this correct column as well with the vector data. And here we're setting data to an array that takes Boolean values, which is will be handled just fine. And we're going to also provide a description here. Make sure when you're specifying custom columns that you have a good description so someone else who's reading this file will understand what those columns mean. And finally, now that we've created this trials object, we can now add it to the NWB file with nwb.intervals_trials equals trials. And I'll go ahead and execute this command. Okay. Okay, so now that we've added some of our behavior information, let's go into the optical physiology. So here we're going to, one, create an imaging plane 2, add acquired 2 photon images. And then 3, add the image segmentation object. So this is for ROIs. And then finally, we can store the information about the fluorescence traces that go with those ROIs. So as I said before, the goal of NWB is to have all of the metadata within the file that somebody else would need to analyze the file. So in that spirit, before we actually write any of the acquired or processed data, we're going to store a little bit of metadata about how the experiment was set up. And we're going to store that in an imaging plane object. This imaging plane object is actually a bit of a misnomer because this can also apply to cases where you're using volumetric imaging. So you can think of it as imaging volume as well. It has a few attributes: name, description, excitation lambda, indicator, location. And it also contains one or more optical channel objects, so we're going to need to create these before we create our imaging plane object. And it also has a reference to a device, and this is where you would store information about your microscope. So first, let's create our optical channel object. It's going to have a description that I'm just going to set to description, and an emission lambda that I'm going to set to 500, just as an example. You can also create your microscope here. So here's the device name I'm setting to microscope. And then I'm creating a device object with a description and a manufacturer. And here I'm setting that device within the NWB object. So that for that you use this general devices attribute, and you set that. You take the device name and you set it to the device value. Now, because this is a reference here, we're going to need to use an object called a soft link here. And this type takes as input the path of the device within the resulting HDF5 NWB file. So that, in this case, is going to be general devices device name. We're going to plug that into our soft link constructor to get our device soft link. Now that we have our device and our optical channel, we can create our imaging plane. The optical channel argument, since it's not a reference or anything, you could just put it directly into the imaging plane. And then we have description, and then our device, we're going to use this soft link that we created. And then we have excitation lambda, imaging rate, indicator, and location. And here, finally, once we've created our imaging plane, we can set that within the NWB object with this command here, where we're setting that name equal to this object within the set. Now that we have our imaging plane, we can move forward with creating our 2 photon series object. So 2 photon series is another type that extends from time series, much like the spatial series that we saw earlier. So just like spatial series, it has a name, a description, a data field, and then it has these 2 different ways of specifying the timing of the samples. In addition, it has an imaging plane field, which is a reference to this imaging plane object that we just created. So in addition, for 2 photon series, there are 2 different ways of specifying the data. You can add the data directly to the NWB HDF5 file, or you can externally link to the data. So I'm going to show you how to do both. So first, because we have a reference to this imaging plane, we're going to need to create that reference using a soft link. So here we have the path of the imaging plane object within the HDF5 NWB file. And now that we have that as a soft link, we can create our 2 photon series object. Here I'm, instead of using timestamps, I'm using a starting time and a rate. I'm specifying some fake data, giving the data the units, and then finally adding in this 2 photon series specific, the reference to the imaging plane. And then finally, we can set this. We're setting it into the acquisition section of the NWB file because we're calling this raw acquired data. So I'm going to go ahead and run this. Okay, now here is an example of how you would add this data if you didn't want to copy that data directly into your NWB file. You can reference an external file with a path, and you can also specify the starting frame of that file, specify the format of it, and again, supply your rate and your starting time. There's a little bit of a hiccup here because to Photon Series, because it inherits from time series, the data attribute is still required. So I'm just going to put a very small NaN placeholder here just so that it passes the verifications. But the external file argument tells you that the data is really in this external file. And then the imaging plane path reference is exactly the same. And I'm going to go ahead and set this as a second to Photon Series into the acquisition section of the file. Okay, now that we've stored our raw data, we can go into storing our processed data. The first thing we need to do is specify the form of the ROIs that we found with a plane segmentation object. So this plane segmentation inherits from dynamic table, just like trials. So it has this optional custom columns, which you can use to add attributes associated with each ROI. For instance, what cell type you think it might be. And then there are additional requirements for the plane segmentation object. You either need to specify an image mask or a pixel mask. So these are two different ways of specifying the form of ROIs. And I'll walk you through imaging mask, which is more common. And then you need to also provide a reference to the imaging plane object, which of course we've already created. So similarly to how the spatial series goes inside of a position object that we saw earlier, here we have this image segmentation which can hold one or more plane segmentation objects. So this is useful if you've tried out different algorithms for detecting ROIs, for instance. Then this image segmentation object is going to go into a OPHYS processing module, which will then go directly into the NWB file. Okay, so first let's make up some fake ROI data. So I'm going to say that our imaging plane is 100 by 100 pixels, and then I'm just going to find a random spot within that plane and make a 10 by 10 patch of ones. The size of the image mask is the size of the imaging plane, and you just indicate with non-zero values where an individual ROI is. And you can use Boolean values or you can use continuous values between 0 and 1 if you have kind of a fuzzy ROI detection algorithm or a probabilistic one. Once we've created these, we can then add them to the plane segmentation dynamic table. Here, I'm not going to add any optional columns just for the sake of simplicity. I'm just going to have the image mask. And this plane segmentation table needs a description. We need to specify the element identifiers, which are going to go from 0 to 19. So there are 20 of them. We have 20 ROIs. And then, of course, the link to the imaging plane. And once we've done that, we can, I'm using a bit of a different syntax here where I'm adding the image plane after the fact. So, so here, again, though, it's created using vector data, setting the data and setting the description. Okay. Now, once we've had our plane segmentation, we can add that to an image segmentation object with the name plane segmentation. And finally, we can create our processing module, which is going to be our OPHYS module. We can set the image segmentation object that we just created within this module and then add the module to the NWB file. So in summary, we have an NWB file that contains the OPHYS processing module, that contains the image segmentation object, that contains the plane segmentation object, that contains the actual ROI data. Okay, so finally we can store the fluorescence traces of the ROIs over time. So here's our schematic of all the types we're going to need for this, but the vast majority of them we've already created. We've already created our plane segmentation and our OPHYS processing module. So we're going to store this data inside of an ROI Response Series, which is yet another type that inherits from the time series type. So of course it also contains the name, description, data, unit, and either rate and starting time or timestamps. In addition, it requires ROIs, which is a dynamic table region that points to the plane segmentation object. So dynamic table regions are a specific type of reference that allows you to reference specific rows of the plane segmentation object. So this allows you to have a matrix of ROI responses where it's expressing them of the data from a subset of the ROIs that were detected. Okay, so in order to create this dynamic table region, first we need to create a type of reference called an object view. And this takes as input the path of the plane segmentation object. Then we are going to create this dynamic table region using this object view as the value of the table key. This also needs a description. We're just going to say all ROIs. And then here we're specifying the data. The data here indicates the rows of the plane segmentation table that you are referring to, and it's 0 indexed here, which might be a bit foreign to MATLAB users. You're starting your counting from 0, and then you're going to the number of ROIs minus 1, and that's going to give us all 20 ROIs, from 0 to 19. And then you also need to transpose this in order to get it into the right form to be stored in NWB. Once we've created this dynamic table region, we can now create our ROI response series. So we have the data units, starting time, the rate and the starting time, and we're just going to set this ROIs key to this ROI table region that we just created. Finally, we can create this fluorescence type, which acts a lot like the position type with respect to spatial series, but here the ROI response series are going inside of the fluorescence type, and that is shown here. And finally, we're adding that fluorescence type to the OPHYS module that we have already created and used for the plane segmentation and image segmentation objects. Okay, now we can set this OPHYS module, if we haven't already, into the NWB file and export the file. And there we go. That will do it for all of the data that we're writing into this file. We have input we've input subject information, behavioral information, raw acquired optical imaging data, as well as processed optical imaging data into this file. So let's go ahead and run in advance of this. run in advance of this. Okay, now I'm just going to show you how to read this data. So to read it, again, you just use read underscore NWB equals NWB read, just this NWB read command, and you input the path. And then you can go ahead and traverse this hierarchy to get the data that you saved. You can do read NWB, inside processing, get the OPHYS processing module, get the fluorescence object, get the ROI Response Series within that, and get the data. But when you do that, you find that you get this data stub object. You may have expected to get the matrix, but you get this object instead. And the reason that we do that is this facilitates lazy reading of the data. So it's often the case with these time series and with other types of objects that the amount of data in here is so large that you wouldn't want to read it all into memory at once. So it provides you this view where you can read specific chunks of the data if you want to. So if you don't want to read a chunk and you want to read all of that data, then you can just use the .load command with no additional arguments on the load command, and you can read all of the data. If you have a lot of data, you may not want to do this. You may want to load only a portion of the data. So there's a way to do this, which is use the same load command, but include these input arguments. So this is an example of going from 1 to 5 in the rows and 1 to 10 in the columns. So this is row start, row end, column end. And this gives you a subset of that data, a smaller chunk. Okay, so this is how you would load the data for an ROI response series, and you can also load this data for your image masks. Here I'm loading that data, all of the data in, and then I'm visualizing just the first ROI, and you can see here that this is basically reasonably what we expected, given that I just kind of chose a random 10 by 10 chunk to say that this is the ROI. Okay, so that about wraps it up for this. One thing I wanted to direct your attention to is this API documentation. So this is really, so what we've shown here is a lot of the commonly used types for optical physiology, but there's a great diversity of different types of data that people are collecting, and a great diversity of different types of data objects within NWB. This just kind of scratches the surface. You can go ahead and look through this, this directory of classes to find the data type that might be useful for your experiment. And if you don't immediately find what would be useful for you, then feel free to send a message on Slack or contact us, the development team directly, or an experienced user, and we'll try to direct you to the type that would be necessary for your data. And if we don't currently have it, then we can help you build an extension. Now you can see here that we have, for instance, a DF over F type, and you can see here that, okay, it contains ROI response series. So you can go ahead and look through this documentation. I also want to direct your attention to other tutorials that are available. If you want to continue using MatNWB, there is an extracellular and an intracellular electrophysiology tutorial. We also have tutorials for PyNWB and Python, and we have tutorials for kind of advanced features within PyNWB, iteratively writing data, writing extensions in PyNWB, and advanced I/O, which helps you finally control how the data that you're writing is expressed on disk.